I was sitting in a coffee shop this morning thinking about Achilles. You remember that myth, right?
Yeah.
The Greek warrior dipped in the river Styx to become invincible.
The ultimate soldier. Except, of course, for that one little spot on his heel where his mother held him.
Exactly. The fatal flaw, the one weakness in what's supposed to be this impenetrable armor. And it got me thinking, we often treat big scientific theories like that. They're presented as, well, bulletproof, case closed.
Especially a theory like Darwinian evolution. I mean, it's the bedrock of modern biology.
Right. But for today's deep dive, we're doing something a bit different. We're looking at a stack of sources, papers, transcripts, lectures that argue there is an Achilles heel in Darwin's theory, and it's a big one. It's genetics.
It is a fascinating twist, isn't it? Because usually genetics is held up as evolution's shield, the ultimate proof. But the materials we have here, mostly from researchers like Dr. Robert Carter and Dr. Donald Batten, they argue the exact opposite.
They're saying genetics is the spear that pierces the armor.
That's the claim. And it really boils down to this distinction we're going to keep coming back to, survival versus arrival.
Okay, what do you mean by that?
Well, they argue that Darwin's theory is pretty good at explaining the survival of the fittest, but it completely fails to explain the arrival of the fittest.
So, how things stick around, but not how they got here in the first place.
Precisely. And our mission today is to unpack why they think that. We're going to look at the operating system of life, the math behind mutation, and the history that's written in our genes.
Before we get to all the high-tech stuff, we have to rewind, right?
Yeah.
Back to 1859. Darwin publishes On the Origin of Species. And I think most people just assume he knew everything we do now, just earlier.
Yeah, that's a common misconception, but it's just not historically accurate. You have to remember, Darwin was working in a total vacuum when it came to inheritance. He was a brilliant observer, of finches and tortoises, but when it came to how traits were passed down, he was guessing.
He didn't know about DNA.
No DNA, no mutations. He didn't even know what a gene was. In fact, his theory for how it all worked was something called pangenesis.
Pangenesis. Sounds like a sci-fi villain. What was it?
It was. Well, it was completely wrong. He thought every organ in your body made these little particles called gemmules, and these gemmules would travel through your blood to your reproductive cells.
So if I, say, lifted weights my whole life.
Your bicep would send big muscle gemmules to your sperm, and your kids would be born stronger.
Wait a second. That's Lamarckism, isn't it? Passing on acquired traits. I thought Darwin was the guy who disproved that.
He eventually overshadowed it, sure, but his own proposed mechanism was very Lamarckian. And here's the really wild part. At the exact same time, Gregor Mendel was in his monastery figuring out the actual laws of genetics.
With the pea plants.
With the pea plants. But Darwin never read his work. So the big question these sources pose is a huge what if. If Darwin had known what we know today about the cell, would he have ever proposed his theory?
Or would he have just looked at the insane complexity and said, no way this happens by accident?
That's the question. And to even begin to answer it, we have to look at what we now know. We have to look at life's operating system.
I love that analogy. You hear DNA is a code so often that it's almost lost its meaning. But looking at Dr. Carter's work, he argues we don't really grasp the sheer density of that information.
It's almost impossible to wrap your head around. Carter uses this analogy. If you took the human genome, just the code from one person, and printed it out, it would fill 850 Bibles.
850?
Yeah.
In one cell?
In the nucleus of one cell, which is only a few millionths of a meter across. It's the densest information storage we know of anywhere. Nothing we've built comes close.
That's incredible. But here's something that's always confused me. We think of DNA as this rock-solid thing, right? We pull it out of fossils. It lasts forever.
That's the ideal, yeah. But chemically, in a living system, it's a disaster waiting to happen. It is so fragile. The sources point out that DNA is constantly being damaged. Oh, everything. Thermal noise, oxidation, radiation. They estimate that a single human cell has to deal with up to 1 million DNA lesions.
Hold on. 1 million per cell, per day?
Yes.
Then how are we not just, dissolving into a puddle of errors? My computer would be toast.
Because you have a repair crew, an army of tiny protein machines, these repair enzymes, that are constantly zipping up and down the DNA, finding damage, cutting it out, and patching it.
Okay, that's amazing.
It is amazing, but it creates a massive logical problem for evolution. It's a classic chicken and egg paradox, the instructions to build those repair enzymes.
Oh, no. They're written in the DNA, aren't they?
They're in the DNA, so you can't have the DNA without the repair crew because it would degrade into soup. but you can't have the repair crew without the DNA to code for them.
They had to show up at the same time, the manual and the mechanic?
It has to be an all-or-nothing system, and for a long time, the evolutionary escape hatch for problems like this was junk DNA.
Right. I remember this from high school biology. Like 98% of our DNA is just evolutionary garbage leftovers.
That was the consensus, yeah. And honestly, the mathematical models of evolution needed that to be true. They needed a huge buffer zone where random mutations could happen without, breaking the whole machine.
And what happened?
A massive research project called ENCODE. And they basically killed the junk DNA idea. They found the vast majority of the genome is biochemically active. It's being used.
So it's not junk, it's doing something.
It's doing a lot. The sources describe it as an RNA supercomputer. All those non-coding regions are the control panel. They're regulating everything.
It's an operating system.
Yep.
And you can't just randomly delete lines of code from Windows and hope for a better computer.
You get the blue screen of death, and Dr. Carter points out it's even more complex because it's four-dimensional.
Okay, walk me through that. 1D is the sequence of letters.
Yeah.
A, T, C, G.
Right. 2D is the network, how different parts talk to each other. 3D is the physical folding. The DNA is folded in such a specific way that genes needed for the same job are brought physically close to each other, even if they're far apart on the linear string.
The shape itself is information.
Absolutely. And 4D is time. The shape changes during development. But here's the real kicker. Overlapping codes.
Like a crossword puzzle.
Exactly like that. A single letter can be part of the code for a protein, part of a regulatory code, and part of a folding instruction, all at the same time.
So if natural selection tries to change that one letter to improve the protein,
You might break two other things. It's a level of data compression that just defies random trial and error.
this leads to the part of the research I found. Well, honestly, kind of unsettling. If the system is this delicate and we're always mutating, what's actually happening to us? The story is we're getting better, right? Ascending.
These sources argue the exact opposite is happening. They call it genetic entropy.
Entropy. Things falling apart.
It's a concept from Dr. John Sanford, a pretty famous geneticist. His argument is that the human genome is essentially rusting out.
Rusting out. That's a bleak image.
It's just math, really.
Yeah.
We accumulate about 100 new mutations per person per generation. So you have 100 more typos in your code than your parents did. Your kids will have 100 more than you.
But isn't that where natural selection is supposed to come in and save the day? The bad mutations get weeded out?
It works for the really catastrophic mutations, sure. If you're born without a heart, that doesn't get passed on. But most mutations are what they call slightly deleterious. Think of it like a tiny new spot of rust on a car.
Okay, one little spot won't stop the car. I can still drive to work.
And because you can still reproduce, natural selection doesn't see it. It's too small to matter for survival right now. It slips under the radar. This idea is called Kimura's box.
Kimura's box.
Right. Selection is like a sieve. It catches the big, deadly rocks. But the slightly bad mutations, they're like sand. They slip right through the mesh. And that sand just piles up generation after generation.
So the argument is that we are, as a species, functionally degrading, losing information.
That's the claim. And it connects to something called Haldane's dilemma. J.B.S. Haldane was a famous evolutionist who did the math on how long it takes to establish a good mutation in a population.
And what do you find?
He found there hasn't been nearly enough time. Our reproduction rate is just too slow. To get from an ape-like ancestor to a human would require fixing millions of new mutations, and the math says we should have gone extinct from the cost of all the bad mutations long before that could ever happen.
Which implies a much younger timeline for humanity.
It points that way, yes. But I can hear people listening and asking, but wait a minute, we see evolution happen? Bacteria, finch beaks?
The peppered moths, right. Are these sources saying that stuff doesn't happen?
No, not at all. In fact, they point out that creationists like Edward Blythe were writing about natural selection before Darwin. They just saw it as a conservative force, a culling mechanism.
Culling, so it removes things, it doesn't create them.
Exactly. It explains the survival of the fittest, not the arrival. If you have wolves with long and short fur and an ice age hits, the short fur wolves die out.
You're left with only long fur wolves. They've adapted.
They've adapted, yes, but they've lost the genetic information for short fur. They are now more specialized, but less genetically diverse. No new information was created.
Okay, but what about a new trait? Like that famous E. coli experiment, Atlenski's experiment, where they evolved the ability to eat citrate. That sounds like a gain.
It's the classic example, but when you look at what actually happened on a molecular level, It's not what you think. They didn't invent a new engine to process citrate.
They didn't.
No. It turns out E. coli already had a citrate pump. It was just switched off when oxygen was around. The mutation was that the switch got broken. It was stuck in the on position.
So it didn't build a new machine, it hot-wired an old one?
That's a great way to put it. It was a beneficial breakage in that one specific lab environment. It's like being on a sinking ship and throwing heavy cargo overboard. It helps you survive, but it doesn't explain how the cargo got there in the first place.
And they say this is a pattern. Stickleback fish losing armor, cavefish losing their eyes.
All broken genes. It's devolution, not evolution. Burning the furniture to stay warm.
So let's pivot to the history written in our genes. We hear about mitochondrial Eve and Y chromosome Adam.
Right. So we know from genetics that all humans trace their maternal line back to one woman and their paternal line back to one man.
And the standard model says they lived hundreds of thousands of years ago. Right. And probably not at the same time.
Correct. But that timeline is based on a molecular clock that is calibrated using the assumption that humans and chimps split about 6 million years ago.
So you're assuming the evolutionary timeline to prove the evolutionary timeline.
It's a bit circular. But the sources we're looking at cite studies that use real-world observed mutation rates, what we actually see happening in families today.
And what happens when you use that clock?
The timeline collapses dramatically. For mitochondrial Eve, it goes from hundreds of thousands of years down to about 6,000 years.
Wow, that's a huge difference.
It's massive, and the diversity patterns seem to fit. Human genetic diversity is incredibly low compared to other animals like chimps.
Which suggests we came from a very small group very recently.
The sources argue it fits a model of a huge population bottleneck, like the flood, and a single dispersal event, like Babel.
What about Neanderthals? I always pictured them as these separate, brutish ape men.
That view has completely changed. We now know they interbred with modern humans. In fact, most people listening have some Neanderthal DNA.
Which means, by definition, we have to be the same species.
Exactly. The argument is they were just another group of humans, an isolated post-flood population. They made art, they buried their dead, they were us.
Okay, we have to hit the big one, the chimp comparison. The 99% similarity, that's the mic drop in any argument about this. Is that number even real?
The sources argue it's incredibly misleading. It's a classic case of cherry-picking the data.
How so?
That 98 or 99% number only comes from comparing the parts of the genome that already align really well. It completely ignores huge sections that don't match up at all, the structural differences, the so-called junk.
So you ignore the differences to show how similar they are.
Pretty much. When researchers like Dr. Jeffrey Tompkins did comparisons of the whole genome, the similarity drops a lot, maybe as low as 70 or 80%.
70%? That's not a cousin, that's a different species entirely.
It's a massive gap. The Y chromosome, for example, is radically different. To get that many changes from a common ancestor in the time allowed is, they argue, mathematically impossible. You're back to Haldane's dilemma again.
So if we try to pull all this together, it's a completely different picture. We have a genome that's like a 4D operating system where the hardware and the software had to show up at the same time.
Great.
We have a mutation rate that suggests we're degrading, rusting out, not evolving up.
Genetic entropy.
We have natural selection acting as a culling process, breaking things to survive, not building new things, and a genetic history that points to a very recent origin from a very small group.
That's the synthesis. And I think that computer analogy really is the key.
What do you mean?
DNA is the hardware. It's the physical molecule. But the code on it, the language, the error checking, the syntax, that's software. And in our entire experience, software never ever writes itself. It always has an author.
A blank hard drive will never spontaneously generate Windows 11 no matter how long you wait.
Precisely. The sources are arguing that you can't have the hardware without the software and vice versa. They had to arrive together, fully formed.
Which points away from random chance and towards, well, intelligent design.
That's the conclusion these scientists come to. They see the genome not as a sloppy record of accidents, but as an engineered masterpiece that's slowly wearing down.
It really flips the whole story. Instead of us being on this upward path to becoming super beings, we're more like a vintage car.
Yeah.
Beautifully designed, but it's got a lot of miles on it, and things are starting to rust.
It does leave you with a really provocative final thought. If the genome is rusting out at a measurable rate, if we really are collecting 100 new errors every generation...
Does that mean we have a species-wide expiration date?
Maybe. But look at it the other way. If we're losing information going forward, that means if you go backward in time, the information gets better, cleaner, more perfect. And it forces you to ask, what was the original genome like? How perfect was that source code before the very first spot of rust appeared?
That's something to think about. We think of the past as primitive, but maybe genetically we're the ones who are running on fumes.
It definitely changes how you view the story of life.
Absolutely. Well, that is a lot to process.
Hmm.
And whether you agree with it or not, it's an argument that's worth understanding. A huge thanks to the work of Dr. Carter, Dr. Batten, and others for the material we looked at today.
It's always good to look at all the evidence.
Thanks for listening to this deep dive. We'll see you next time.